Welcome to another edition of Research in the Sciences, a set of methods videos for students by students. Today's episode, Experimental Designing, Making or Breaking the Experiment.
Hello and welcome to our laboratory. I'm Callie.
And I'm David.
I'm Parker. Today we will be teaching you about experimental design and each component of it.
That's right. Experimental design represents an essential element to every experiment and ensures that there will be an outcome that can be measured effectively.
But what exactly goes into making an experimental design? Do you know the basics?
Great question. But before I answer it, let's take a few short moments to check out what we currently know about experimental design. Pause this video now and write down all the components you can think of that make up an experimental design.
Great. Now that you've written down the basics of an experimental design, we can build upon that and ensure that you know everything you need to know to conduct good science.
Most importantly, we will help you to link experimental designs to the right statistics that will help make your data more valuable and usable.
Correctamundo. David, good job on linking design to statistics.
Thank you.
Specifically, our video will help you achieve the following objectives. 1. Learn what constitutes an experimental design. 2. Understand how to successfully create an experimental design. And 3. Correctly assign appropriate statistical tests for factorial experiments.
Thanks, Parker. So let's get right into it. First, we need to talk about what actually goes into an experimental design, and here are the basics. A basic factorial experimental design includes factors, levels, treatments, replicates, and an appropriate statistical analysis. I know that's a lot to remember. We have created a useful mnemonic to help you remember. Falters, factors, and levels make up treatments, which exhibit replicates, and statistics make it easy to remember the order of components of an effective experimental design. So remember, science never falters with good experimental designs.
Well, that's certainly helpful.
Thank you.
But what do all these categories really mean? Well, in short, factors refer to independent variables comprised of multiple levels. A combination of levels and factors results in treatments. Scientists then replicate these treatments multiple times to produce data sufficient for analysis through specific statistical approaches appropriately related to the number of factors. It all connects.
Now, let's break down some statistics. We will focus on tests that look at means, also known as averages. To choose a statistical test, you need to figure out how many groups and factors you have. A t-test only works for 2 groups, but if you have more than 2 groups, you need to use an ANOVA, or analysis of variance. To determine which type of ANOVA to use, you count your number of factors. For example, if you have 2 factors, you use a 2-way ANOVA.
Let's say you get a job in a pet store that specializes in carrying lizards, and you want to make them as happy as possible, so you do a little experiment to see how temperature affects lizard activity. You have 10 lizards and 10 tanks and want to decide between 2 temperatures, 30 degrees Celsius and 35 degrees Celsius. Ask yourself these questions. How many factors are there? How many levels are there? How many treatments should you have? And how many replicates should you also have? In this case, you should have identified temperature as the only factor of interest. And with one factor, levels and treatments are the same. Yours are 2 temperatures, 30 degrees Celsius and 35 degrees Celsius, and you have 5 lizards to watch their behavior at each temperature. That makes up your replication. Now the important part. Because there are only 2 groups and 1 factor is being tested, we would use a t-test to test if the lizards bobbed their heads or performed any other behavior the same number of times at the 2 different temperatures.
Now let's modify this example. What if we set up an experiment with even more tanks that vary in temperature? We randomly select lizards to be placed initially in each temperature area, and the temperatures include 30, 32.5, 35, 37.5, and 40 degrees Celsius. Again, ask yourself these questions. How many factors are there? How many levels are there? How many treatments are there? And how many replicates? Still just one factor, temperature, but now 5 levels or treatments, 30, 32.5, 35, 37.5, and 40 degrees Celsius. So now we still only have one factor, but we have more than 2 groups. Because we have more than 2 groups, we can no longer use a t-test to tell the difference between groups. A one-way ANOVA would have to be used so we do not compound type 1 error. See some other method videos if you still want to learn more about this. Therefore, in this case, we would also want to do a multiple comparison test that tests the differences between treatments.
Let's do one more example. What if we use 2 different species of lizards the pet store just got in a shipment of 2 sets of 20 individuals, and then set up an experiment that has tanks with 2 different temperatures? Once more, ask yourself these questions. How many factors are there? How many levels are there? How many treatments are there? And how many replicates are there? How many factors are there? In this case, 2 species of lizards and temperature. How many levels? 4, 2 for each. That makes 4 treatments. Species A at 35 degrees Celsius, species B at 35 degrees Celsius, species A at 40 degrees Celsius, and species B at 40 degrees Celsius. And I have 40 lizards, so n equals 10. Because there are 2 factors, the proper statistical test would be a 2-way ANOVA. No post hoc test needed because only 2 levels per factor.
Now let's test your understanding of these concepts with one more pet shop scenario.
Just one more time, ask yourself, how many factors are there? How many levels are there? How many treatments are there? And how many replicates would you recommend? Take a minute to sketch out an experimental design.
I hope that you identified factors as the type of food and temperature. In this case, we only have one type of lizard, so that is not a factor. Our levels consist of 3 levels of food, food sticks, crickets, and mealworms, and 2 levels of temperature, 35 degrees Celsius and 40 degrees Celsius. To get treatments, you simply multiply the 2 levels together to get 6 treatments. How many replications did you suggest? Replications, or the number of trials done per treatment, should at least equal or even double the amount of treatments.
Great work on experimental design.
Thanks.
What did you suggest for the statistics?
Always ask yourself, are there more than 2 groups for factors or levels? If there are more than 2 levels, you need to be clever and run additional tests to determine which independent variables are causing the significant differences. The correct approach involves a 2-way ANOVA. Remember, you have 2 factors and then a post hoc for type of food because you need to see if a significant difference exists between food sticks, crickets, and mealworms, the 3 levels of that factor. Even though you have only 2 temperatures, a t-test does not work because you have more than 1 factor and more than 2 groups total. Now, that's easier to understand. If you still feel confused about what constitutes an experimental design, ask for help or even review our example. 2x3 experimental designs commonly occur in biology research due to ease of analysis, so understanding a basic experiment within that model will prepare you better for the world beyond this video.
So remember, science never falters with good experimental design. Following the parts of falters also helps you choose an appropriate statistical analysis. Remember, falters, factors, and levels make up treatments which exhibit replicates and statistics.
Now you know what constitutes an experimental design, falters, how to successfully create an experimental design, replication, when to use appropriate statistical tests for factorial experiments, ANOVAs.
The why is obvious. Good designs make for good science.
We hope that our video really helped you with your understanding of how to develop an excellent experimental design. Thanks so much for watching Research in the Sciences, Making or Breaking the Experiment.